So I'll go back and repeat what I said. So that's what we're recording now. So your bone marrow, okay, is your main hematopoiesis tissue, okay? Your spleen and your liver can also do some hematopoiesis. So the term hematopoiesis basically means to form cells, okay, to form blood cells, all right? So this occurs in your bone marrow all the time. So your bone marrow, your red marrow, we actually have two types of marrow. We have a yellow marrow and a red marrow. The red marrow is what does your actual producing your red blood cells, okay? When we're really, really little, up till about 12 years old, most of our bones, long bones especially, have marrow. They have red marrow, okay? As we get older, 12 to an adulthood, the red marrow is actually replaced with yellow marrow. Yellow is just like lipids, fats, those kind of things. And just the ends of the long bones, so we talk about long bones, we're talking about like your humerus, your femur, the radius, the ulna, you know, those kind of things. Right on the ends of those bones, you have red marrow, okay? And we'll talk more about this tomorrow in lecture, but just to go ahead and give you an idea of what's going on. So anytime we're talking about the marrow, it's just referred to as the bone marrow, okay? We have cells that's where they form, and they're baby cells. They're immature. They should always stay in that bone marrow. They should never be in the peripheral blood, okay? So we said peripheral blood is the blood that's in our veins and in our arteries, okay? So anytime you hear me talk about blood smears, peripheral blood is what we're talking about, okay? Or if I say something about in circulation, okay? Those cells are in circulation. Then I'm talking about peripheral blood, all right? They're circulating through your body, all right? So what we're going to do today is we're going to look at the 5 cells, white blood cells, that you see normally in the peripheral blood, okay? Now, later on, we're going to discuss those immature cells that should always be in the bone marrow, okay? But sometimes when there's abnormalities like diseases, things like leukemias, even some severe anemias, those cells that should normally stay in the bone marrow get kicked out early into the peripheral blood. And that's that, I guess, trigger or clue that we've got something going on in the bone marrow, okay, with that patient, some disease state that is happening in that particular patient, okay? So eventually you guys will be able to identify, okay, what's called a blast and then the lineage of each cell type, okay? But today we just want to focus on normal white blood cells that you're going to come in contact with every time you do a diff. Does that make sense? And when I talk about a diff, I'm talking about a differential, all right? So you're going to take a slide. You're going to look at it on 100 on the oil immersion, and you're going to go in a certain pattern, and I'll show you the pattern, and you're going to count 100 white blood cells, okay? And you're going to differentiate those 100 white blood cells, and that's what we call a diff, all right? So you don't pick and choose which cells you want to go. 100 white blood cells, you identify that cell, and then you look at that percentage of that particular cell line. And based off of which one's elevated or which one's low, gives the doctor the idea of what's going on with that patient, if there's an abnormal white blood cell count, okay? And so, but you've got to be able to identify the cells to be able to do that, okay? You may not get this little flow sheet I made, okay? All right, now I know you're looking at this and you're thinking, well, dang, that's pretty simple, okay? But I'm telling you, it really is that simple. If you will learn this and get this in your brain, you'll never have a problem identifying a cell, okay? Now listen to me. Cells don't read textbooks, okay? Does that make sense? You may come across a cell that don't fit this, all right? But if you'll look at it long enough and go through these steps and decide what it is, you will get it right, okay? It doesn't necessarily mean it's going to be the picture perfect cell, but it will fit the category. Does that make sense to you? All right. Okay. All right. Any questions so far? All right. So let's look at the cells. All right. Most common cell. All right. This is the most common cell that you're going to see on a diff normally. Okay. It is known as a neutrophil. All right. The neutrophil is going to make up about 60, I'm going to say 65 to about 73%. Okay? I know that sounds crazy. And it may not be exactly that, but that's going to be pretty close. Okay? All right, so what does that mean? So that means when I do a 100 cell differential, I should count between 68 and 73 neutrophils. Does that make sense? Okay? Now, neutrophils, all right, make up the majority of your cells. What is their job? Their function is phagocytosis. You ever heard that term? Phagocytosis. Okay? It basically means they Pac-Man stuff. You know what Pac-Man is, right? Everybody knows what Pac-Man is, no matter how old you are, right? Pac-Man. So they eat things. They engulf it. Okay? Phagocytosis is a term. It's also known as endocytosis. You may have heard that in biology. Okay? So basically they go and they're a little amoeboid cells. So they are mobile cells. And they engulf foreign particles. Okay? This can be bacteria, it can be yeast, it can be fungi, it can be parasites, it can be red blood cells. It can actually be another cell. Hello.
Hello, this is Sarah from Discover. I am calling in regards to rate expiration. We've been trying to reach you as your eligibility for rate reduction is about to expire.
Don't have time to mess with you today, or I would. Okay? Alright, so, neutrophils. Alright? So, notice on your sheet at the top, first two boxes. Okay? It asks if it's a granulocyte or an agranulocyte. Alright? So look, we have two types of granules. We have what are known as primary granules and secondary granules. Okay? Alright, so think back to biology. Okay? So when you were in biology, you should have learned somewhere about organelles inside a cell, right? One of the organelles, or actually two types of organelles that are common in all cells, are known as lysosomes and peroxisomes. Y'all remember that from biology? Okay? Basically, they're little vesicle-filled granules. And they contain enzymes or special proteins that help them to do jobs, okay, depending on what it is. Things like lysosomes and peroxisomes, they do things like kill bacteria, okay? Lysol, lysosomes. Same thing, I promise, okay? So, granules are important to us, okay? Because we know that if a cell has a granule in it, okay, and you can see those little dots in them, okay? Those are granules, all right? If we see that, we know automatically that it's one of three cells, okay? It's going to be a neutrophil, an eosinophil, or a basophil, okay? So the next thing we have to do is we have to determine what kind of granules that it has, okay? Neutrophils always have small granules, that's key, small, tiny granules that are usually blue and pink, okay? Now I'm going to give you a buzz term. Y'all know what a buzz term is? You ever heard that term? A buzz term is something that's associated with something that you could use to describe it, okay? So, neutrophils, the cytoplasm is described as salmon color, okay? Y'all know what a salmon is? Salmon, how do y'all say it? I don't know. Everybody out here it multiple ways. But the fish, you know what I'm talking about? Pink, blue color. And they refer to it as a salmon color in general, okay? Tiny, multicolor, pink and blue granules.
And which one is that?
The neutrophil. That's what we're talking about. You got it? Yeah. We're talking about the neutrophil right now, okay? Tiny, pink, blue granules, okay? Keyword, tiny, all right? Because the next two that we're going to look at, they have granules, but they only have one type of granule, okay? Neutrophils have both. Have both primary and secondary granules, okay? They have both types of granules. So that's the reason we have blues and pinks inside there, okay? Because they have both types of granules, all right? Questions about that? Okay. So as in the bone marrow, okay, we have a particular cell that's called a myeloblast. You don't have to know that right now, but I want to make a point, okay? So notice the nucleus of this cell is all broken up, okay? All right? So in the bone marrow, this cell has one solid mononuclear nucleus, okay? And as it grows, all right, it goes through stages where the nucleus kind of gets a little smaller, all right? And then as it goes, the nucleus kind of gets smaller. And then eventually, it's going to get this shape, okay? And then eventually, it's going to get smaller, and it's going to form a horseshoe shape, okay? Now, we're going to talk about this one in just a second, okay? Because here, this direction, should be always bone marrow, not bowel movement, okay? Don't get that BM mixed up. They're two totally different, okay? All of these guys should always be in the bone marrow. Now listen, you're going to learn each one of these later on, okay? So I'm not really worried about it at this point, okay? But this one, and then the one like on the board that you're seeing here, can be in the peripheral blood, okay? These two can normally be in the peripheral blood, all right? I always use this analogy. This guy is known as a band neutrophil, okay? Because it looks like I guess a rubber band or something. I don't know, but it's called a band, okay? They were actually used to be called stabs. A long time ago, they were called stabs. Matter of fact, when you guys get down in the lab, we have these little counters that you use that you have to push the button and it keeps up with a total. And it has a picture of all the cells on it, and it's actually called a stab. The band is called a stab. All right? It's called a stab. All right? But this one is called a segmented. And by its name, the nucleus is segmented, okay? At this point, it's still one nucleus. It's just getting skinnier before it breaks down, or before it becomes segmented. All right, so this guy, the analogy that I always use, this is the 17-year-old who can work, he can do the job, but he's just not real good at it. Does that make sense? So sometimes you got to keep the 17-year-old out of the house and make them help pay the bills. You know what I mean? Same way. So like when you get a really, really bad infection, okay, you're going to see your body respond by kicking out some earlier cells out of the bone marrow. Because they can work, they're just not real good at it, okay? They're not as good as this guy, okay? But this guy can only do so much, and if the infection's really, really bad, then he has to have some help. Does that make sense? Okay? And sometimes these two can get kicked out of the bone marrow also in really, really bad infections. And we'll talk about that later on. Okay? These two guys should never be in the peripheral blood. Okay? They should always stay in the bone marrow. If you ever see these two in peripheral blood, we're dealing with probably a leukemia. Does that make sense to you? But sometimes these guys can get kicked out a little early, especially this guy. He's known as a metamyelocyte, and we'll talk about that later on. Okay? All right, but this two right here, I'm going to show you pictures of. Okay? All right. Now, is this guy still a neutrophil? Yes, he is. So it's still going to look just like your picture there with the pink and blue granules. Okay? It's just his nucleus is going to be kind of horseshoe shaped instead of being broken up, like I said. Okay? Does that make sense? All right? This is a segmented neutrophil. Okay? Questions about a segmented neutrophil? Okay? Main function, phagocytosis. So in other words, you see an increase in bacterial infections. Okay? Fungal infections. Those kind of things. Anything that needs to be engulfed, you're going to see an increase in this guy. Okay? Does that make sense? Alright? So if I ask you a question later on, what kind of infection do I see an increase in neutrophils? What are you going to tell me? Bacterial. Okay? That's very, very important. Alright? And I'm pretty sure Ms. Cole's probably already mentioned this cell to you guys, considering I've just had microbiology. Okay? Sometimes they're called, let me get you this term. Neutrophils can also be referred to as PMNs. Okay? You might see that term. It's morphonuclear cell is what they refer to it as. It's a newer term that's starting to be used. But most time in here, we'll always use neutrophil. Okay? Segmented neutrophil, band neutrophil, those kind of things. Does that make sense? Okay? But sometimes in your book, you may see PMN. Alright? So just be aware that it's the same thing. Alright. So this little description is very similar to what I just told you guys. Okay? I'm not going to stand up here and read it to you though. You can read and I'm going to post this for you guys. Does that make sense? Okay? Questions about a seg? Anybody? Good? All right. All right. Seg. I've got some pictures. A few more. Okay. These are all segs. Okay. So, pink, blue granules. Right? I see them. Okay. Nucleus is broken up. Chromatin's rough. What we call rough chromatin. It's always going to be really, really dark. You see little wrinkles in it. Okay. Because this cell is mature. All right. It's not growing anymore. This cell, we're going to talk about this pretty soon, like tomorrow or Monday, depending on how far we get. Cells reach a certain point where they don't go through mitosis anymore in your white blood cells. Okay? So like the blast that I was talking about earlier, okay, and then it's the pro-myelocyte. Myelocyte. And then it's called a myelocyte. And and then it's called a metamyocyte. And then you got that band, and then you got the seg, okay? These guys, from here back to this point, can go through mitosis, okay? So what does that mean? That means for every blast, I can do multiple divisions through mitosis. Does that make sense? Okay? And then as they're maturing, then this one can do multiple divisions and go through it. Okay? And then, of course, the same for this one and the same for this one. But once we reach this stage, this is the last stage that it can do any kind of mitosis. All right? Once we get the band and the seg, that's it, no more division. So what you get is that mature cell that's not going to divide anymore, and it'll eventually die. That makes sense? But as long as these guys are still in the bone marrow, they can continue to produce cells that we need, okay? And then, of course, this one would go to become a seg, this one, and so forth. And that's the reason we can continue to have our white blood cells. Does that make sense? Okay? So when you start seeing cells like this, you know it's mature because now the nucleus is broken up. So of course it can't do mitosis because what's necessary for mitosis? What you gotta have? An intact nucleus with what? What's inside the nucleus? DNA, right? You have to have an intact set of DNA to make sure that your cell can continue to do what it's supposed to do, right? And in this case, we don't. Our DNA is all scattered out and broken up, and it can't reform and form another cell. Does that make sense? Okay? All right, so these are neutrophils. These are the things you can see. Notice the different shapes of the nucleus, okay? There's no way to ever determine what the nucleus shape is going to be. But you can look at the granules, look at the cytoplasm, and then look at the nucleus being broken up, and you can know if it's a neutrophil or not. Does that make sense? Okay. Questions about a neutrophil? And here's some other pictures. Okay. See, you got weird ones, and you even got smiley face ones. I've seen heart ones, I've seen rabbit ones, seen them that look like a penis. I'm just saying, okay? You just don't ever freaking know what you're going to get, what they look like. But they will always have the same characteristics. Does that make sense? They're going to have granules. They're going to be pink and blue granules. They're going to be tiny granules. And then the nucleus is going to be usually broken up, okay? That's what makes it a seg, a neutrophil. Okay? Questions about that? Good? All right. So, this is the mature seg. This is what you're going to see most of the time on your normal diffs. 65 to 73% of the cells that you come in contact with on a 100-cell diff are going to be neutrophils. Okay? And they're going to be mostly segs. Does that make sense? You may see 0 to 5 bands. Okay? Which is this cell, okay? So your band neutrophil, okay? Same characteristics as the segmented neutrophil with the exception of the nucleus. The nucleus is still intact, okay? All right, so, Mr. Thomas, what makes a band, okay? My rule, this is my rule, and it's important because on cell division, remember what I told you guys? Who made the choice of what the cell is? Me, right? Okay? So, me, I like for the band, or the nucleus, to be about 50% all the way around. Okay? In other words, I want it to be about the same all the way around. Okay? So look, so if you get something like this, that's not 50%. So to me, that's not a band. Okay? This is already going to seg for me personally. Okay? Alright? Here's why. I know y'all think I'm crazy. Okay? When in doubt, listen to me close. When in doubt about a cell, okay, call it the more mature cell. That makes sense? Okay? Because if you start calling a bunch of immature cells, then guess what's going to happen? The doctor's going to start ordering crap that might not necessarily be needed. Does that make sense? So, one of the big things that we see an increase in bands is pneumonia. Okay? Patients who have bacterial or bacterial most of the time, bacterial pneumonia, you see a huge increase in the bands. Okay? And that's a thing that doctors look for on a diff. If they see more than 5 or 6 bands, then they start ordering chest x-rays and everything else. Does that make sense? And so it can ultimately cost your patient more money than what's really necessary. Okay? Because you called too many bands. Does that make sense to you guys? Okay? So bands need to be really, really uniform before I call them a band. Okay? They got to be just about horseshoe shape. Okay? Now listen. This guy, right, is also band shaped. Okay? I'm going to go ahead and show you, draw it. Okay? It is going to be a cell that looks like this, though. And it's going to have a little bit of cleavage right there. Y'all laugh at me, that's what it's called. It's called cleavage. Okay? It cleaves. In other words, it's starting to go toward that band stage. Okay? It looks like a kidney shape. Does that make sense? If you see a kidney shape, then it is always a metamyelocyte. Okay? So it's not a band anymore. So for a band, it needs to be pretty narrow, but it also needs to be an even band. Does that make sense? Okay. Questions about that? All right. So it does the same thing as a neutrophil. Just remember, it's just the baby neutrophil. It's the 17-year-old. It got kicked out of the bone marrow a little earlier than what was necessary. Does that make sense? Questions about that, guys? All right. Got a few more pictures of bands. All right. So this one, I don't like this picture. This was actually from the Cellovision site. Actually, I got this off of it. I don't like this one. This one I would probably call a metamyelocyte if I was calling it myself. Because see, it's barely got just a little bit of cleavage, and to me that's really, really kidney shaped. And the only thing that I can think of that the reason they didn't call it a metamyelocyte in general is because it's a little smaller than a metamyelocyte in general, the whole cell. Because one of the things we'll talk about, they're large to smaller. Okay? Just a general rule, as cells mature, they get smaller. Does that make sense? Okay? And I know you are thinking, well, Mr. Thomas, how the crap can I measure stuff? I don't have a measuring tape in my microscope. You see these little critters? What are these? They're red blood cells. Okay? I'm going to give you ways of looking at red cells, normal red cells, and give you an idea how many it takes to cover a cell normally. Okay? For instance, the lymphocytes that we're going to talk about in just a minute. The way you can determine a lymphocyte from a monocyte, other than just the cytoplasm that I've got on there, is the size of the nucleus. The nucleus of a lymphocyte is about the size of a red cell. You could take a red cell and cover the nucleus, just the nucleus, not the cell itself, but the nucleus of a lymphocyte. And that lets you know that it's not a monocyte. Because a monocyte, you'd be able to put 4 or 5 red cells on top of it. Does that make sense to you? Okay. And we'll talk about that later on. But to me, if I was doing the diff, that guy would have been a metamyocyte. It wouldn't have been a band. Does that make sense? All right, questions about a band? All right, here's a couple more pictures. Okay, so this one's a little bigger, but then it's still pretty even across there. So I would have called those bands, both of those bands, if I was counting them. Okay, that makes sense? All right. What about the color? Still salmon? Yeah, still got granules, small tiny granules, you know. Probably what the deal is, this stain, the pH is probably off a little bit on the pH. But other than that, it still has all the same characteristics of a band. Does that make sense? Okay, and that's the key. In your brain, right now, today, in your brain, you need to learn that flow chart. Okay, you come across a cell, every cell, if you don't know it for sure, go through this flow chart. Has it got granules? Does it not have granules? Okay, if it's got granules, then I know it's got to be one of 3. Then I start looking at those granules. Okay, the granules will give the cell away. All right. No matter, listen to me close. No matter what's happening with the nucleus, okay, you don't even have to have a nucleus. Does that make sense? If you'll learn the granules, because they're always the same in these cells. All right, that makes sense? Okay. Now the agranulocytes, the monos and lymphs, they can be confusing sometimes, but I promise by the time we get done, you guys will know them. Okay. All right, questions about the neutrophils, segs, bands. All right, those are the most common ones you're going to see. All right. Okay. Now, jot down somewhere bands, 0 to 5% can be normal. You know, it's not an abnormal thing to see 0 to 5% of those bands. Okay. But usually not more than 5. You're usually not going to see more than 5 bands on a diff. All right. Questions about anything, guys? All right. So, next granulocyte. Okay. This is my Auburn cell. That's what I call an Auburn cell. It is orange and blue. Okay. Everything. Now listen, you're going to hear people say red, all right, red granules. But I promise you, they are usually orange, okay, because of the way the stains are, all right. Eosinophil, okay. Eosin this literally translates into acid lover, okay. So what that means is the granules that are in this cell like the acid of the stain, okay? So they're going to be red. Acids usually stain red. Eosin is red, okay? Orange, all right? Red, orange, all right? Now, look at the size of the granules compared to the neutrophil. They are huge. They look like boulders inside the cytoplasm compared to those little primary and secondary granules of the neutrophil. Does that make sense? Okay? EOs are most always, not all the time, but most always bi-lobed. Okay? That means they're going to have 2 lobes. Does that make sense? Okay? Not always, but like 90% of the time, they're bi-lobed. All right? They'll usually only have 2 lobes of the nucleus. That makes sense? But what did I tell you a while ago? Do we have to look at the nucleus? No, we do not, because all we have to do is look at the granules. The granules will always tell us it's an EO. All right? Very, very unique looking cell. All right? EOs, 0 to 5% of your diff. All right, so I'm going to tell you something that's going to be really, really confusing. Okay? This cell and this cell, you can't tell what it is at that point. Okay? But this cell and this cell will eventually turn into either a neutrophil, an EO, or a base cell. Okay? I know that sounds crazy, but just trust me. Okay? But once it reaches this stage, you will be able to take it. Does that make sense? You can have an eosinophil myelocyte, you can have a basophilic myelocyte, and then you can have a neutrophilic myelocyte. Okay? I know that doesn't mean anything to you right now, but later on it will. Okay? So I'm trying to make you understand it today that this cell, depending on what cytokines are chemicals that are attached to the cell that make it do things. Okay? So they did help differentiate this cell into one of these three. Okay? And the reason I'm telling you that is because the other two, lymphocytes and monocytes, have their own lineage. Does that make sense? Okay? So we have what's called a lymphoblast and a monoblast. Okay? Unfortunately, this one and this one, you can't tell the difference in. Monoblast and myeloblast look exactly the same. Okay? Morphologically. Okay? We're going to talk about this thing that we can do now, technology-wise. It's called flow cytometry, where we can introduce antibodies that are labeled for certain antigens that are on the cell membrane, and it'll help us determine if it's a monoblast or a myeloblast. Does that make sense? If it doesn't, don't worry about it. I always get ahead of the game because it's fun. This is a fun class. Okay? I love hematology. Hematology is my thing. I like it a lot. Okay? All right. EOs. So what do EOs do? Okay? You see an increase in EOs in two things. Allergies, parasitic infections. Okay? So if you have an increase in EOs, it's usually going to be in the springtime. Okay? When people's allergies are acting up, and sometimes even in the fall because of hay fever, ragweed. Okay? All right? Okay. Off subject. See how smart you guys are. Okay? Ragweed. What's it look like? Huh? I knew you was going to say that. Who else says that? Yellow flowers, sides of the road, yellow, right? And that's ragweed. Wrong. Did y'all know that? That is goldenrod. It's a plant of its own. There's actually a plant that grows right beside goldenrod. It is green. It doesn't ever change color. It's got like green flowers, green leaves, everything. That's ragweed. But who gets blamed for every year? Everybody. Goldenrod. Everybody blames goldenrod for their allergies, and they call it ragweed. But it's not. It's a totally different plant. Did y'all know that? You did know that. You know there's a ragweed and a goldenrod. Okay. Well, I thought you was blaming goldenrod for ragweed. Okay. So she knew it. See, I'm actually a botanist. like my master's degree, I did a lot of plant stuff. And so like, I'm like qualified to teach like botany and things. And I didn't know that until, of course, I went through those classes. Because I was brought up that that yellow stuff that's ragweed. You know, that's what's causing everybody to be sick and everything else. But it's really not. There's two plants growing really, really close to each other. And it's actually the green, ugly plant
Do
that's
they always
causing.
grow beside each other?
Well, not necessarily, but usually they're in the same fields and that on the sides of the road and that kind of thing. Yeah. So, but I figured everybody would say the yellow ragweed, because that's what everybody usually says.
Ditch lilies too.
Huh?
Ditch lilies.
Yeah, yeah. Talking about the white stuff,
The little orange flowers.
the orange ones. Yeah,
Call them ditch lilies. They're lilies.
yeah. But that's just daylilies. They just grow
in the ditches.
sometimes in the ditches. It's, a lot of times it's probably because somebody may have had a house there at one time. You know. All right. Anyway, off subject. Sorry. Well, not kind of off subject, but not really. So secondary cytoplasm abundant contains eosinophilic, which means orange. They're secondary granules. Okay. So like in the neutrophil, the pinker ones that you see, those are also secondary granules. Okay. So these guys though are really, really large, and they contain histamines. Okay. The granules, so like neutrophils contains like lysosomes and peroxisomes. One of the things that's in the granules of eos is histamines, which is what we need to trigger our allergies. Okay. All right. They also have some of those granules contain things to help destroy parasites. Okay. So roundworms, you hadn't got there yet. Have y'all had lecture for Miss Cole yet? Y'all are still doing bacteria though, right? Y'all still got a few bacteriology stuff to do. So the next chapter you guys will do is fungi, I think. Y'all doing fungi next, or y'all going to start parasites? Did she say?
She hasn't decided.
She hadn't decided yet. Anyway,
What'd you say that destroyed?
parasites. Yeah, parasites. All kinds of parasites. Roundworms especially. So things like ascaris, tapeworms, malaria, any kind of parasite that you get in your body, you're going to see an increase in those eosinophils because that's their job. Okay? All right, questions about this? No, it's a lot, but I promise you'll get it as we go. Okay? But the big deal for today is the pictures. Make sure you know the pictures. Okay? All right, here's some more EOs. Okay? This one here is the typical EO. All right, see that bilobed there? Which this one's bilobed too, it's just got a little extra, a couple of segs, okay? This one's not as common, but most of the time they're gonna be like this shape, shape 2 and 3, okay? All right, questions about that? All right, so the next granule site is a basophil, okay? Basophil literally means basic lover, okay? So the stain that it's going to take is the basic side of the stain, the higher pH of the stain. So it always gives a blue color, okay? See the really, really blue granules? And as a matter of fact, the granules are so large that you can't tell much about the nucleus, okay? Basophils, very, very large granules. Okay, purple granules, black granules, ever how you want to look at it, okay? They're going to be really, really large, purple, black granules, okay? All right? Their function is anaphylaxis and inflammation are the two big things that you see the increase in basophils. Anaphylaxis, what's anaphylaxis? Extreme allergic reaction, like you win, like you're gonna die reaction. Okay? So anaphylaxis and inflammation. Inflammation is actually the main thing that we see because anaphylaxis is actually pretty rare, right? But inflammation is not. So if I ask you the most common cause of an increase in basophils, then you would say inflammation. Okay? Does that make sense? All right? So, basophils, purple granules, okay? These guys all come from the same precursor cell, the myeloblast. Myeloblast, promyelocyte. When we get to the myelocyte stage, we can tell if it's an EO, baso, or neutrophil. Okay? That makes sense? All right. One important thing I need to tell you guys right now, before we go any further. All right? So we see this cool picture of a basophil on the screen, right? Got all the characteristics, big purple granules, broken up nucleus, those kind of things. All right? When you're doing your slides, they're not going to be like this. There's only going to be a few of the granules in it, but they will still always be blue. Here's why. The granules of basophils are water soluble. So guess what happens to stains a lot of times? They get watered down. Okay? Just from the rinse process, a lot of water ends up getting in the stain. And so what happens is you dip that slide in that stain that's got water in it, and the water dissolves the granules. So you usually only see really tiny blue granules, but there'll be some big ones in there to give you away. Okay? You won't have that salmon color. The pink ones won't be there. Does that make sense? And that'll help you to determine that it's a basophil and not just a neutrophil. All right? Basophils. Basophils, 0 to 1%. Okay? So it is normal to do a diff and not ever see a basophil. Okay? And it's abnormal to see more than one. Okay? So 0 to 1%. All right. Questions about the granulocytes. 3 granulocytes: neutrophils, eosinophils, basophils. Okay? Questions? Good? Good with those. All right. So let's move on. So our first agranulous, well, here's some cells. You can see how there's only a few granules in these, and the reason it is because the water probably dissolved some of them. Okay? So this is what you typically see, like this. Instead of that one, it's got a bunch of granules in it. This is typical. This is what you're typically going to see. Okay? Now you can barely see the nucleus, but this nucleus is segmented, just like a neutrophil. Okay? It's just the granules are usually so large that you don't see the nucleus as well. Does that make sense? Okay? Questions about the basophil? All right. First, agranular cell. Okay? So what does the term agranular mean? It doesn't have any granules, right? So that's a Q, correct? All right. The first one is a lymphocyte. Okay? First one is a lymphocyte. Now I know, okay, this says small lymphocyte. There are actually 2 types of lymphocytes in our body. There's actually 3. But there's actually 2 that you see most common. You have T cells, and then you have B cells. Okay? T cells tend to be like that one. Small. The nucleus to cytoplasm ratio is high. Okay? In other words, you just barely see a little bit of cytoplasm because the nucleus takes over the entire cell. Just about. Okay? Alright? Lymphocyte cytoplasm is usually blue. Okay? Now you can see these weird ones where it's kind of a light blue, almost white. Okay? But 9 times out of 10 it's going to be blue. Alright? Okay? It's not going to have any granules in it. Also, the nuclear to cytoplasm ratio. So you're going to see this sometimes. N to C. That means nucleus to cytoplasm ratio. Okay? Alright? So, nucleus to cytoplasm ratio is going to be high. In other words, the nucleus is increased compared to the cytoplasm. Okay? NC ratio. Alright? These are usually the T cells. So the cell that you're looking at on the screen is a T cell. Okay? T lymphocytes here. Alright? The chromatin is a little bit rough, but it's still got some smoothness to it. Okay? Alright? So earlier we talked about size. Remember I told you guys. So a normal lymphocyte can actually help us determine if these red cells are normal, micro, or macro. Okay? I know that sounds crazy. Alright? But a red cell should be able to lay right on top of the nucleus of a lymphocyte. That makes sense? Okay? If it can fit on top of a lymphocyte, then it is a normal red cell, known as a normocyte. Okay? If it is smaller, it is a microcyte. Okay? And if it's larger, it is a macrocyte. And we'll talk about that later on when we start talking about the red blood cell stuff. But those are the things that help us to determine size of the red cell. Okay. All right. So what do lymphocytes do? Okay. T cells, these are our immunity. Okay. So what T cells do is T cells actually attack viruses. Okay. So these are going to be the ones that are activated to destroy viruses. They're going to make little chemicals that can kill viruses. Does that make sense? Okay? This is known as your, always get this mixed up. This is your cell-mediated immunity. Okay? It's called your cell-mediated immunity. Okay? There are types of T cells. T4, T8, and then you have what's called the natural killers. Okay? So, people who have HIV, the HIV virus, the virus that causes AIDS, the HIV virus destroys the T4s and the T8s. That's what makes them immunocompromised. Yes, ma'am?
So when you say like Q4, like is that like related to like thyroid?
No. The T4 is actually a compound that's it's called thyroxine. Okay, it has 4 iodine molecules in it. Okay, it is your main thyroid hormone. Okay, we look at things like free T4 and T4 levels, but no, that's totally different. This one, the reason we call it a T4 is because if we could like do a flow cytometry, there is a certain antigen on the outside of the membrane that determines that it's a T4. Okay, and then a T8 is the same way. There's another one that's got certain shape that makes it a T8. Okay, but these guys are what's responsible for your immunity. Okay. So you remember yesterday in lecture I was talking about the little boy with the bubble? Okay? This was his problem. His body didn't produce lymphocytes. And so he didn't have any kind of an immunity. Okay? Alright, so that's the T cells, the small lymphocytes. Okay? Any questions about those? Alright, so you see what they look like. Okay? Mononuclear, agranular. No granules in them. Okay? Tiny, smaller, about the size of a red cell. It's always going to be a lymphocyte. Okay? Alright, here's a couple more pictures. Notice the cytoplasm's a little larger in some of these, but guess what they're missing in the cytoplasm? Granules. There are no granules. This one right here kind of looks weird, like it has some granules, but those are not true granules. Those are known as azure-philic granules. They're usually red in color, but look at the cytoplasm, how blue it is. Okay? And we know from the granulocytes that they have to be salmon color, orange, or what? Purple, right? And what's this color? Blue. So we know it's not one of those granulocytes. So it has to be either a lymphocyte or a monocyte. Does that make sense? Okay. All right, so these are all lymphocytes, different forms of lymphocytes. And most likely, okay, now listen to me closely. When you're doing your diffs, you don't have to determine large lymph or small lymph. You just call it a lymphocyte. Okay? Because in a second I'm going to show you another cell that they call a large lymphocyte. It's actually a B cell. We'll talk about that in just a minute. But when you're doing your diff, you don't have to distinguish those. Okay? All right, so lymphs are our second largest cell. And I'm going to say 30 to 40%. Now, it may be off a little bit, but that's pretty daggone close. Okay? Is that okay with you? And actually, some books may actually have it 20 to 30. It just depends on your population. Okay? All right. One thing I do want to make mention. Okay? Remember yesterday in class we talked about reference ranges? Remember? Talked about that. I showed you in the book your reference ranges. And I said a couple of things affect reference ranges. What were they? Location, age, sex, ethnicity, those kind of things, right? Okay? Children from birth to about 12, their neutrophils and lymphocytes are reversed. They tend to have more lymphocytes normally than neutrophils. Does that make sense? So like they may have 60 to 70% lymphocytes and neutrophils may be only 30 or 40%. Does that make sense?
Is that kind of what kids usually be a little bit more sick than adults?
Well, yeah, because kids' immunities are developing when they're young. So yeah, kids do get sick more. Plus kids are at school all the time. You know, so they're always around other kids. And other kids are sick. You know what I mean? But yes, that's why. So when they're a baby, they're trying to build their immunity so their body produces more lymphocytes. Okay, so tomorrow in class, the chapter that we're going to discuss is about hematopoiesis, but it's about the organs that produce hematopoiesis. So we're going to talk about how babies in the womb use the yolk sac, and then they go to their liver, and then eventually they go to bone marrow. Okay? Whereas adults, we go to bone marrow. That's our big one. Okay? But bone marrow produces cells, but lymphocytes don't mature in the bone marrow. They actually go to another organ called the thymus. Okay? The thymus gland in us is about that big. I mean, it's barely detectable. Okay? Whereas in a baby, it's this big. You know? It's like a butterfly looking shape, big piece of tissue in their chest. Okay? Around their throat area. Okay? The reason it is because the immunity is so low at that point, their body produces more lymphocytes. And so therefore their thymus is where their lymphocytes mature. So the more they got, the more tissue they're going to have. Does that make sense? Whereas the older we are, our immunity gets established. And so our thymus actually begins to shrink because it's not needed anymore as much. Okay? We still always have it, but it's very, very tiny and doesn't produce like it should. Okay? Because then we get the thymus and then we have the lymph nodes and that kind of thing. And we'll talk more about that tomorrow. Okay? But lymphocytes. So T lymphocytes are usually small cells. Okay? But remember what I said, on the diff, it's just a lymphocyte. Make sense? Okay. That's all you got to know, that it is a lymphocyte. All right, so they're calling this one a large lymphocyte. And you can notice it is a lot larger, but if you lay a red cell on top of it, the nucleus is still about the same size as a red cell. Does that make sense? Okay. The difference is the NC ratio. And this one, the NC ratio is about even in these larger cells. All right. If we did flow cytometry, This cell would probably be a B cell, okay? B cells are unique, okay? Because in our bone marrow, we can actually have some B cells. Even though most of them go to the thymus, just like the T cells, lymphocytes, you can keep some B cells in your bone marrow. Very few. All right. The reason we do is because the B cells. Okay. So we said this one is your cell-mediated immunity, right? This one is known as your humoral immunity. Okay, so what does that mean? That means that these guys, the cells themselves, do the actual killing. So they're cell-mediated, okay? So the lymphocytes go out, they come in contact with the virus or whatever foreign material it is, they produce chemicals that destroy it, okay? These guys mature from a B cell to another cell that we'll talk about later on called a plasma cell, okay? Then that plasma cell maturates into what we call immunoglobulins. All right, what's another name for immunoglobulins? Anybody know? Antibodies. Okay, so how many of you guys got vaccinated for COVID? Got the flu vaccine, you got your MMR vaccine, whatever you had to have as a vaccine as kids, right? So what a vaccine does, it stimulates B cells, okay? So you put that foreign particle in your body, whether it's a virus or whatever, and your body says, okay, B cells, you need to learn, memorize what this virus is, okay? And then you program yourself that if it ever comes in my body again, you're going to make antibodies to it. Does that make sense? That's humoral immunity, okay? B cells die, but they also go through mitosis. And so they divide and make daughter cells that are known as memory cells. And those memory cells are what helps us stay vaccines to work the way they're supposed to, okay? Because vaccines don't really, we don't want this guy just going in and killing it, because it might do that and it might kill the virus, but then the next time the virus comes, it doesn't matter, because the B cells didn't do their job. Does that make sense? So we have no memory of the virus. And so we can't produce antibodies to fight it off, okay? So what we did, or what they did with the COVID vaccine, is they created a little piece of messenger RNA and injected it into you, and then your body said, okay, that's foreign. And so it stimulated one of these B cells to make an antibody to destroy it, okay? So then that B cell becomes a memory cell, and then the next time COVID goes into your body, okay, what are viruses? What are all viruses? They're either one of three things. What are they? They're either DNA, RNA, or protein. That's it. They have no other organelles. They don't have a nucleus. They don't have anything like a bacteria or a real cell. Because you guys know viruses are not living particles, right? They're considered non-living things. They can't reproduce on their own. They have to have a host cell, okay? So really all a virus is just a ball of DNA or RNA and proteins. Some of them are proteins. They're kind of weird, but they are proteins. But the majority of them are that. They're either RNA or DNA. So, RNA, right there. We took a piece of RNA out of that virus, created a vaccine with it, okay? So now when that virus gets in our body, our B cells over here sense it, and they come in, and the antibodies attach to it and die. Starts producing antibodies. Turns the B cell into a plasma cell turns into antibodies, and then they go and those antibodies attach to the virus. And what it is, and the one for COVID, is it's attaching to that spike protein. The spikes on the virus itself are made out of protein. So there's a piece of RNA in there that codes for that protein. And so as soon as that virus gets in there, it senses that code or that protein, it goes and makes antibodies to it. And those antibodies attach to the virus and destroy it. Won't allow it to replicate like it's supposed to. Okay? So actually, I know everybody's, oh, I'm not taking a vaccine and all this. This vaccine, scientifically, is probably the best vaccine that we've ever created. You know that? Because how are other vaccines made? What do they use? That's it. They use either a dead or a weakened live virus. It just depends. The COVID vaccine has no virus at all. It's using its internal structures to kill it. So it's very, very specific to this particular virus. So in science, so you get the politics out of the way, and the science is probably the best vaccine we've ever created. Okay? Everybody pitched a fit, and listen, I was skeptical. I really was. When all this crap started in March of in 2020, I was like, this is a bunch of bull crap, it's just political, it's stupid. All of it, because 99% of the people were surviving who were getting it, so it wasn't like it was that big of a deal. But then I got it, and it about killed me, So I realized that it wasn't. So, but it did get political. It really did. But people like us, who are science people, don't listen to politics. You look at the science behind it, the research. Okay? So everybody was saying, oh, they made it too quick and had, put it on the shelf too quick. You didn't have the studies and all this other stuff like you normally do. That's bullcrap, because actually this technology has been around for like 20 years, but nobody would ever fund it to do it. And then when COVID came along, they were desperate, and the scientists said, hey, we've got the technology that we've been doing for 20 years, and none of y'all give us money for it. So the government said, we'll give you money. And that's the reason they were able to push it out so quick. Does that make sense? Okay. All right, so B cells, large lymphocytes, they look just like the small lymphocyte. Structure-wise, everything else are just a little larger, okay? But if you look at the cells, okay, blue cytoplasm, no granules, rough chromatin of the nucleus, it still has to fall into the lymphocyte category. Does that make sense? Okay? All right. These are different ones, but they're all lymphocytes. Everybody agree? Okay? Okay, the next agranulocyte, and the last cell that you're going to come in contact with, it is known as a monocyte. Okay? Known as a monocyte because it has one mononuclear nucleus. Okay? All right. Largest cell in the peripheral blood. Okay? They're huge. You can lay 3 or 4 red cells on top of it. Okay? And you could lay 2 or 3 on top of the nucleus of it. Okay? All right. Buzzword. All right, like I said a while ago, buzzwords. Buzzword for monocytes. Ground glass cytoplasm. Okay? Literally, sometimes they look like you took glass and ground it up and put it in the cytoplasm. Okay? The cytoplasm is usually a gray color. Okay? And if you look, it looks like it's got little tiny granules in it. That's the ground glass that I'm talking about. Okay? Those are not granules. They're not true granules. Okay? The next thing that I use as a buzzword, you'll never see in a book, it has a butt nucleus. Okay? A the nucleus looks like a butt. It actually is bent over, looks like two butt cheeks. Okay? I promise. Alright? The next thing that's unique about monocytes is vacuoles. Okay? So go back to biology. What is vacuoles? They're storage areas. So they're holes, right? But you can put stuff in them. So think about what monocytes do for their function. Their function is phagocytosis, just like neutrophils. Okay? So they're going to have some vacuoles to store stuff. Does that make sense? It's kind of not scientifically correct, but analogy is good. Okay? Vacuoles are storage places. So if you see vacuoles in a cell, they're active cells. Okay? Because later on, when we get a little further in, neutrophils can sometimes have vacuoles. Okay? So people that have really, really bad infections, you'll see their neutrophils have vacuoles in them too sometimes. Okay? And the whole point is because of phagocytosis. Alright? Because of them become reactive. Alright? So monocytes, large, gray, ground glass cytoplasm, really, really large nucleus that sometimes looks like a butt. Okay? Questions? Alright? Function, monocytes. Function, phagocytosis. Alright? Monocytes don't stay in the peripheral blood very long. Okay? They actually only stay in there a matter of a couple of hours. Okay? 0 to 10% of your diff can be monocytes. 0 to 10% can be monocytes. Okay? Monocytes are known as macrophages when they leave the peripheral blood. Okay? Monocytes are known as macrophages. Macrophages. And they usually don't stay in the blood very long. Okay, so a couple of hours, and then they usually migrate into tissues or body fluids. Okay, so things like paracentesis fluid, which is fluid in your abdomen, pleural fluid in your lungs, fluid that's in your pericardium, which is the sac around your heart, synovial fluid, even like fluid in your joints. All these places tend to have a predominant of macrophages as their cell types, and we'll get into body fluids. There's a whole separate thing, of course, for body fluids. But you're going to see macrophages, but just keep in your mind that macrophages are monocytes. Monocytes in the blood, macrophages in the tissues, but it's the exact same cell. Okay? They just get bigger as they get older. Okay? Questions about that? Okay. All right, so here's the different pictures of monocytes. Okay, so make sure you know these. This one, like I was talking about, that looks like a butt, doesn't it? Yep. And I promise you, you'll see them a lot like that. That's how they're kind of shaped. The nucleus is kind of shaped like a butt. Okay. Ground glass, so you can see it's kind of a grayish looking color compared to the blue of a lymphocyte. Okay. And in just a minute, I've got a slide that's got a monocyte and a lymphocyte side by side, so that you can see the difference in the two. Okay. All right. Questions about monos? All right. Here we go. All right. What's this? What's this? Monocyte. See the difference? Okay. So you can definitely tell. Smooth nucleus, one big intact blue cytoplasm, no granules, right? So agranular, blue cytoplasm, lymphocyte, almost always, okay? All right. Monocyte, large, gray cytoplasm, ground glass cytoplasm, but nucleus. And look how the nucleus is a little rougher looking than the lymphocyte. Okay? See what I'm talking about by rougher? See the little dark areas? That's called rough chromatin of the. So like the chromatin is the DNA when it's come unwound and it's just kind of packed inside the nucleus. Okay? Lymphocyte, monocyte. Okay? B cell, T cell. Doesn't matter, does it? Right? Is that not what I told you? Doesn't matter. What's it called? It's a lymphocyte. It's all matters right now. Okay? When we get later on, we start talking about flow cytometry and those kind of things, then it'll matter. But right now, just a lymphocyte. That's all you need to know. Okay? Questions? All right? Lymphocyte? Lymphocyte? Sure. What do you think, Jordan? You shaking your head no? They look what? Look different. Look purple, don't they? Look at the cytoplasm, guys. See that? See that? That's different than that, isn't it? Totally different, right? They are different, okay? Remember I told you earlier that sometimes cells look alike, but we have to go through our little sheet, okay? So, yes, they're agranular, but what's the next step? Cytoplasm what? Blue to white blue, right? Blue-white kind of, right? What's these? That blue? Purple, isn't it? Kind of purplish blue, correct? It's a big difference between that and those, right? What about size? It's huge, right? That's a lot bigger than that cell, isn't it? What about this one? Lot what? Smaller than that one, right? Okay? It's a trick, okay? These are nucleated red blood cells. These are actually should always be in the bone marrow. You should never see these, okay? These are not even white cells. These are red blood cells. These are immature red blood cells that should be in the bone marrow all the time, okay? But sometimes people have anemia, they get kicked out of the bone marrow early, okay? And sometimes you may see them in the peripheral blood, but you've got to be able to recognize that that and that don't look the same. Does that make sense? Okay? Another thing, look at the nucleus. It's almost perfectly round, where this one has got some little curves in it, little cleavage and stuff. All right, what about the chromatin? See how rough that chromatin is? It's like a brain, almost, right? This one's still kind of smooth. It's got some darkness in it, but it's not clumped up like this one, okay? And then this one up here doesn't even look anything like it because of the cytoplasm and that, okay? This is known as a rubocyte. So this is like almost the last maturation of a red cell, okay? This is known as a metarubocyte. This one is about to lose its nucleus, and it's going to be a red cell, okay, pretty soon, all right? So we hadn't talked about that yet, but I wanted to see if you could tell the difference in the two, okay? And listen, and this is very, very important, okay? You know what hematology is, really? Is being able to determine what's normal and what's abnormal. Sometimes it doesn't matter if it's abnormal, as long as you can know that it's abnormal. And the only way you can tell something's abnormal is by knowing what? What's normal, okay? And that's the key to hematology, is knowing what's normal, what normal always looks like, by those characteristics that I've given you. And as long as you know that what's normal as soon as you see something that's abnormal, your brain will automatically say, hey, that doesn't look right. Does that make sense? And then guess what? There's these guys and women that work in the hospital that have went to school for, let's see, 4, 8, 12, 16 years and get paid about $450,000 a year to determine what that abnormal is. And they're known as pathologists. Okay? So you don't ever have to make the final call if something's abnormal, okay, or what it is. All you have to do is be able to determine that's not normal. And then you go and you pull out a little piece of paper and you write path review. And you set the slide on it and you take it back to the pathologist. And then they look at it and they say, hey, you did a good job. That's definitely abnormal. Those are myeloblasts, or those are promyelocytes, or those are nucleated red cells, whatever. Okay? Does that make sense? So the important thing is to know what's normal. And then when you see abnormal, you'll automatically know that, hey, that doesn't look right. And that's my biggest goal for you as a tech or a student, is to know what's normal so that you can recognize abnormal. Okay? Now, I do expect you to know the abnormal, the common abnormal stuff. In other words, if I show you a blast cell, I want you to know that it's a blast by its characteristics. They have specific characteristics that you'll learn, and you'll know obviously what it is. Okay? And you'll know that, hey, that don't belong in the peripheral blood. Okay? And then you put it up for path review. Does that make sense? Okay. All right, so nucleated red cells, what those two are, very common, very similar to lymphocytes, but they are not. Okay? All right, what's this cell? Okay, first step, what do? Granules or agranule? Granular for sure. So, now my next step is to look at what? Color of the what? The granules. What are they? Purple. So what are they? So what's my choice? Salmon, orange, red, purple. Right. This one's purple, so what's it got to be? Thank you. Hard? Ain't hard about that? Nope. All right. What about it? Granular, agranular. Granular. So what's my next thing to look at? Color of the granules, right? Salmon, red, orange, purple. Which one is it? Red, orange, so it has to be a eosinophil. Every single freaking time. Okay? Makes sense? Okay? And it is bilobed, so that helps you too. And the picture's a little bit blurry, right? But you got enough information to say, hey, that's an EO. Right? Okay? Agranular, granular. Granular for sure. Right? Alright? Pink purple.
Pink blue.
Orange red. Or big purple. So salmon. Right? So it's a what? Neutrophil. What kind of neutrophil? Is it a seg or a band? That one is a band. Good horseshoe. Pretty even all the way around. Right? I know it's got a little bit of a crease there, but not, that's still less than 50% of the total. Right? Okay? That makes sense? Band. Right? Neutrophil. Band. What did I say the old name for it was? Stab. There you go. Very good. Y'all guys been listening. All right. What about it? Granules? Agranular? Well, let's think about the granules. Okay? Are they salmon, big orange, big red, big purple? None of those, right? So we're going to say it's agranule because that's what they have to be granules. They have to be pink and blue, two kinds. They have to be big orange or they have to be big purple. So it's definitely not granular, right? So what do I do? A granular. So then what's my next step? What am I going to look at? Color. Is it blue? Is it gray ground glass? Gray ground glass. What's the nucleus look like? Right? So guess what that is? It's a monocyte. Make sense? Listen, I know y'all think, well, God, he's being goofy. Do it every time. If you will go through your mind that flow chart, you will get it right every single time, because even though they don't read books, they still have the same characteristics every single time. Okay? They have to. And if not, put it up for a path review. Right? Because it's not normal, because those are normal things. What I've got you on that sheet are normal things that they're all going to have. Okay? All right. Granular? Agranular. Granular. Salmon? Big orange? Big purple. Salmon. Seg or band? Seg. Right? Neutrophil. Segment and neutrophil. Any problems with that? Okay, good. Saw somebody go, Okay? Granular, agranular. Agranular, right? Alright, so cytoplasm. Blue or white? Gray or ground glass? Gray or ground glass. I know it looks a little bit of tinge blue, but it's not blue. And plus, it's got that ground glass look. Okay? What else does it have? Vacuoles. Okay? 9 times out of 10, if vacuoles are present, it's going, and it's big like this, it's going to be a monocyte. Okay? Alright? And my nucleus looks like a butt. Okay? What about size-wise? Large. Right? You put 3 or 4 red cells on top of that thing. Correct? Monocyte. Every single time. Okay? Alright, what about this guy? Lymphocyte. Agranular, blue-white. Okay? The little stain's not really as good. Okay? Blue-white, though, definitely not ground glass. No vacuoles. No butt nucleus. So, hey, it's got to be a lymphocyte. No matter what. Correct? There you go. All right, so listen to me. Thursday, I'm going to post this today for you guys. Thursday, you will have a little small practical on just cells. Okay? Be just like what we just did. All right? I'll say, what is it? All right? And you just tell me what it is. Make sense? All right, guys. We did stay a whole hour, didn't we? Stayed an hour and a half. Y'all talk too much.